enzymes which can break and seal the DNA strand. You can see there are 4 options and all the options are nothing but enzymes. Let us start with the first one, topoisomerase. Topoisomerase 2 is the enzyme which is having the capacity or which is capable of breaking the DNA, what we call give a nick and then again join it. Helicase is another enzyme which is involved in DNA replication. It helps in unwinding of the 2 strands of DNA. Primase. Primase is basically an RNA synthesizing enzyme. REN, what we call restriction endonuclease. It is an enzyme which is involved in biotechnology for cutting the DNA in the restriction sites. The right answer over here is option A, topoisomerase 2. Next, cistron is. Let me first tell you what a cistron is. A segment of DNA which can produce a specific polypeptide chain is called a cistron. The options are coding sequence of DNA. Coding sequences of DNA are called exons. Now the next 2 options, intervening non-coding sequences of DNA and sequences that are removed during RNA splicing. These are nothing but interons. Since these do not code for any proteins, during RNA splicing, the sequences which do not code for any genetic information, they are basically removed. So these 2 options are also wrong. The next one, functional unit of DNA molecule that codes gene product, and this is the right answer. The next question, the structural genes of lac operon transcribe messenger RNA which is. Now you are aware of what a cistron is. Cistron is nothing but a segment of DNA which can produce a specific polypeptide chain. So basically, if you take a RNA, in case of eukaryotes, it is found that the RNA does not get transcribed completely. The one which is required, only that part of the messenger RNA gets transcribed. That's why we say monocistronic. Mono stands for single. So one cistron is transcribed at a time. Whereas the first option, polycistronic, means more than one or many cistrons are transcribed at a time. So in case of prokaryotes, it is found that because there are no interons in between, all the sequences present are coding for proteins. It is found that the transcription starts from one end and it completes till the end, as a result of which you can see a polycistronic structure is found in prokaryotes. Now you can see replicative. Replication is just a property of DNA. Monokaryotic. Karyon means nucleus. Mono, single. So cells which have a single nucleus is called monokaryotic. So here the right option over here is polycistronic. So you are aware of the example lac operon, where it has got 3 structural genes, gene Z, gene Y and gene A. So when the transcription goes on, it is seen that all the 3 genes are transcribed at a time. So let us solve the next question. If a double-stranded DNA has 20% thymine, the percentage of guanine in the DNA is 30%, 10%, 90% and 40%. We are aware that nitrogen bases are of two types. Purine and pyrimidine. Adenine and guanine are purines. Thymine and cytosine are pyrimidines. According to Chargaff's rule, purine always pairs with pyrimidines, and the base pairing is also very specific, that is A always pairs with T and G always pairs with C. So here if you see, the percentage of thymine given is 20. So what will be the percentage of A? It has to be 20. So thymine and adenine together, it forms 40. C plus G is equal to 100 minus 40. This gives 60. Now G plus C, if it is 60, then the percentage of G is equal to 60 divided by 2, that is 30%. So the right answer is A. Students, let's see the next question. It's match the following. Match the entries in column 1 with those of column 2 and choose the correct answer. You can see here all this information which is given is with respect to DNA fingerprinting. What is actually DNA fingerprinting? DNA fingerprinting is basically an analytic technique to identify individuals based on specific nucleotide sequence present in an individual. So here you can see the different steps which are present, or the different steps of DNA fingerprinting. To begin with, actually DNA is collected. If the amount of DNA is not sufficient, then they are subject to PCR, that is polymerase chain reaction, wherein the amount of DNA is amplified. Later it is treated with REN, that is restriction endonuclease. These enzymes cut the DNA into small fragments. Once they are cut into small fragments, they are subjected to electrophoresis. This is a technique where the fragments of DNA gets separated on the basis of its size on a gel slab or an agarose gel slab. Once they get separated, they are treated with alkali. Why are these DNA fragments treated with alkali? This is because the double stranded DNA has to be converted into single strand, or the double strands have to be separated so that single strands are available. Why this is done is because the next step what comes is DNA probing, where synthetic DNA probes are added. So those probes, they have to go and bind to the DNA. So DNA has to be initially separated into single strands. Later they are subjected to another technique which is called southern blotting. Here the fragments which are present on the gel slab, they have to be transferred to a nitrocellulose sheet. So these are basically the steps that you find in DNA fingerprinting. Let us match it with the explanation which is given in column 2. Alkali treatment. So here this is basically done to split the DNA fragments into single strands. Southern blotting. This is a technique where the DNA fragments are transferred to nitrocellulose sheet. Electrophoresis. Here DNA fragments are separated on a gel slab based on its size. PCR, polymerase chain reaction, is for amplifying DNA. Autoradiography. This forms the last step of DNA fingerprinting, where the DNA fragments are exposed to X-ray films so that barcode like film is obtained, or barcode like structures are obtained, which will be later compared with the fingerprints, and an individual will be identified. So, autoradiography is nothing but exposing the DNA fragments to X-ray photographic plates. Then DNA treated with REN. This is basically done so that the DNA can be split into small fragments. So, based on this match, you can see the right answer over here is A matches with N, then B matches with O, C matches with M, you can see the option 4, D matches with R, E matches with P, and F matches with Q. So, the right answer is option 4.